Okay, so in this video we will talk about enzymes, photosynthesis, and cellular respiration as part of, as part of AP Biology Unit 3. And this video includes a discussion of the enduring understandings and essential knowledges listed in the framework from College Board. All right, here we go. So we're going to start with enzymes, where it says the structure of enzymes includes the active site that specifically interacts with substrate molecules. So in a review of enzymes, enzymes are proteins that fold up in a particular shape dependent on the polypeptide sequence as well as the R groups and their properties. And the folding up of that protein will create a section called the active site. And that active site is where the substrate attaches, and that is where the reaction occurs. Enzymes are biological catalysts that speed up chemical reactions by lowering the activation energy, and that happens when you form an enzyme substrate complex. So in the GIF going on right here, you see, oh, I love this. You see the water is being used in the reaction. This is called hydrolysis. This is splitting a disaccharide into two monosaccharides. So here this is sucrose, which is the white table sugar. And you can also see here how the enzyme slightly changes shape. That is called induced fit. So the structure of enzymes includes the active site that, oh, is that the same standard? Okay, so I also wanted to point out that when we talk about that polypeptide, the sequence of amino acids that forms the enzyme, that is dependent on the DNA sequence. So our genes code for proteins, and our proteins give us our phenotypes. One type of protein are enzymes. So how an enzyme's shape is formed is dependent on the sequence of DNA coding for particular amino acids. So if, for example, there is a mutation in a gene that could possibly change the amino acid and the folding up and shape of an enzyme. But if a gene is intact and is the normal sequence, then how that protein folds should create an active site that is complementary to a substrate. Okay. When we look at how this works, I like to use the analogy if they fit like a lock and key, the active site and the substrate. But really, it's the shape and the charge of the enzyme and the substrate are compatible, which means that like in that active site, if there are, I think I have another slide, sorry. So I kind of like, oh, maybe I don't. But the charges of the amino acids that line the active site should be like chemically compatible with the substrate. So if you have like a polar like active site region, you wouldn't have a nonpolar substrate. Like those would repel each other. Now, the way enzymes work is that they catalyze reactions by lowering the activation energy in cells. Okay, and sometimes an enzyme can be affected by its environment when the environment maybe heats up too high or has certain ions in like salts or a change in pH. It could actually disrupt the bonds in that polypeptide, leading to the unfolding of that protein, and we call that denature. So if an enzyme loses its shape, it denatures, and therefore it can no longer catalyze reactions. So environmental temperatures, so as you increase a temperature, that can lead to increased like molecular movements. But at the same time, eventually a temperature that's too high will disrupt bonds, and then like hydrogen bonds and ionic bonds and different interactions, the molecular like moving of the molecule becomes too great and it unravels or denatures. But at the same time, sometimes changing the pH can also have an impact on the structure of a protein. So when you think about pH, pH is a measure of hydrogen ions in the environment. So a low pH, an acidic pH, has a lot of hydrogen ions. So if an area becomes more acidic, basically you're adding more and more H pluses or protons or hydrogen ions, and therefore that's disrupting the hydrogen bonds as well as the ionic bonds in a folded protein, and that could lead to it denaturing. Okay, so sometimes though, if an enzyme denatures, it could be reversible. So, okay. And this environmental pH, this is kind of a tricky picture. I just liked it because it kind of showed pluses in the environment and ions in the environment. The green lines here represent hydrogen bonds, and how sometimes, like, if you change the ions in the environment, you could end up disrupting those hydrogen bonds and the structure of that protein. Relative concentrations of substrates and products determine how efficiently an enzyme reaction proceeds. So if you think about, I really like this next slide here, where if we look at these different test tubes and you see how there is a constant amount of enzyme. In this picture, the blue represents the enzyme, but as you increase the amount of substrates, you can see how there's a greater likelihood that a substrate will interact with an active site, forming products. So as you increase the substrate, the rate of the reaction increases, the amount of products increases, until you get to a certain concentration or a certain amount where it eventually levels off. So if you look at the data, it goes 1, 2, 4, and then 8, 8, 8. That's because in this example, the enzyme has become the limiting factor. It doesn't matter if you continually increase the substrate. There's a limited amount of enzymes to catalyze the reaction. So therefore, you get a plateau or a leveling off. And that same would be true if you switch that. If you had a limited amount or like a constant input of substrate, but you slowly increase the reaction of or the amount of enzyme, you would get a same shape of graph. Okay. Higher environmental temperatures increase the speed of the movement of the molecules in a solution, increasing the frequency of collisions. So that's why you see that like gradual increase in the rate of the reaction, because as they move faster, the chances of a substrate attaching to an or colliding with an active site also increases. However, eventually you get to a certain temperature where it's just so hot and the enzyme denatures. Ooh, we also have inhibitors. I love inhibitors. I have a whole video on it. But when you look at ways to inhibit enzymes or prevent an enzyme from working, you have two options. There are competitive inhibitors or allosteric or non-competitive inhibitors. And in a competitive inhibitor, it is competing with the substrate for the active site, versus a non-competitive inhibitor is going to attach allosterically or an area that is not the active site. And when you can see here in the green, when a non-competitive inhibitor attaches to the enzyme, it changes the shape of the enzyme so the substrate can no longer fit and there's no longer a reaction occurring. All right, so now moving into, now if that was too fast of a review on enzymes for you, I believe I have 3 separate videos on my Unit 3 playlist that go into detail on enzymes. So the remainder of this video will now focus on first photosynthesis and then cellular respiration. So as we move into living systems, we all require a constant input of energy. So whether we're autotrophs or heterotrophs, we all require energy, and without energy, that results in death. And so this part C right here really ties into ecology or Unit 8 when we think about energy flow in an ecosystem. Okay, so let's go ahead and look at energy-related pathways. That can be super complicated, but I love this picture because it shows different metabolic pathways. So a pathway is basically like a series of steps or a sequential, like controlled series of reactions, all using enzymes. Now, it is an efficient way to like transfer energy, so that way we don't spontaneously combust. And I like the end of this standard right here where it says a product of a reaction in a metabolic pathway is generally the reaction for the next step in the pathway. So let's go ahead and see in general. So here you start with a substrate, and in a pathway, you're going to have enzymes that control each step. And you start with something. So for example, in glycolysis, you start with glucose, and then you end with 2-pyruvates. But it took 10 reactions. It took 10 different enzymatic steps to make that happen. So each step in a metabolic pathway is controlled by enzymes. And the metabolic pathways that we will talk about in the light reaction, in photosynthesis, we have the light reaction is a metabolic pathway. And then the Krebs cycle, the Calvin cycle is a metabolic pathway in photosynthesis. Then when we look at cellular respiration, we have glycolysis, the Krebs or citric acid cycle, and oxidative phosphorylation or the electron transport chain, as well as fermentation. So we have quite a few metabolic pathways we're going to discuss, but what they all have in common is they all rely on enzymes. So first, let's talk about photosynthesis. And photosynthesis is what autotrophs use to capture solar energy and convert it into chemical or usable energy for life. So photosynthesis first evolved in prokaryotic organisms or cyanobacteria, which is like a blue-green bacteria. And so if you're reading a question, a prompt or something about cyanobacteria, those are photosynthetic prokaryotes, and they were responsible for the first photosynthesis on Earth. Is it like 500 million years ago, maybe more? And they oxygenated our atmosphere. Before photosynthesis evolved, our atmosphere was rich in carbon dioxide and low in oxygen. Now, these prokaryotic photosynthetic pathways were the foundation for eukaryotic photosynthesis. And we know today that our chloroplasts evolved from ancient prokaryotes. So kind of rad. All right, so let's go ahead and talk about the light-dependent reactions of photosynthesis. And when we look at this, the big idea is we're taking light energy or solar energy and converting it into chemical or usable energy in the form of ATP and NADPH, which will then be invested in the anabolic pathway of the Calvin cycle to produce our organic molecules, carbohydrates, lipids, proteins, and nucleic acids. So in the light reaction, we can see here a couple different things that we're going to talk about in more detail. So we have the light reaction happens we saw in Unit 2, it occurs in the thylakoid membrane, and we rely on proteins called photosystems, and that is what we're going to look at next. So here we have the light reaction, and light travels in like a photon. It's both a wave and a particle. And we have these areas called photosystems 1 and photosystems 2. And photosystems is PS2 right here, as well as PS1. Oh, I have a pen. Duh. So here you have your PS2. This is a photosystem made of pigments, or contains pigments. So pigments are the molecules, they are proteins, that are going to absorb and reflect light. So if you ever see a graph called an absorption spectrum that looks something like this, like a photosynthesis, usually on the side over here, you have the absorbance. So, and then down here you would have like the wavelength of light. Oh my gosh, Mary. That's being absorbed. And so this is a measure of how much light is being absorbed. And if it's like a high peak, that means there's lots of photosynthesis going on, lots of light being absorbed. If the line is low, that means there's like a low amount of light. That's probably in the green pigment, a green wavelength of light. Okay, so these photosystems though, PS2 and PS1, are what are going to absorb that solar energy. So you can see here, and then that is where electrons will be donated from. So this is the photosystem, this whole thing right here. In the center of the photosystem, we have the chlorophyll A is the main pigment in photosystem 2, and then you have chlorophyll B in photosystem 1. But when the light shines on, you're going to have electrons, which are these orange things here. that are going to get passed into the electron transport chain. So if I go back to this video, this here, the light shines down, electrons are donated, or they're excited to a higher energy state, and they enter into the electron transport chain. Now, in order to replace those electrons, let me clear the ink. As the electrons break free from chlorophyll and enter into the electron transport chain, ultimately, the final electron acceptor is NADP+. It'll gain the electrons at the end right here, becoming reduced to form NADPH. This NADPH is what will transfer over or carry that energy or electrons over to the Calvin cycle. However, we need to replace these electrons so it can continue. That is where the reactant of water is required. Water will be split into oxygen, hydrogens, and electrons. So if we go back to this GIF here, that's what what we're seeing is we are seeing water molecules come in. Watch, almost, here we go. So you're going to see water molecules, come in right here, and then you see them split. The electrons enter, and now you have some, I don't know, maybe this is the hydrogens, this is the oxygen, O2. So when we look at these photosystems, this splitting of water is called photolysis. And it replaces the electrons that were donated into the electron transport chain. Now over here in PS1, these electrons also break free from chlorophyll and will be picked up or gained by NADP+, but it's the electrons flowing down the electron transport chain that are going to replace those right here. So as we travel, or as the electrons travel down the electron transport chain, another thing that happens is that protons are pumped, so it's not diffusion, are pumped into this thylakoid space. So the thylakoid is made out of a lipid or surrounded by a lipid bilayer, just like we saw in Unit 2. And this lipid bilayer prevents protons from diffusing out. And so therefore, or because of this nonpolar center here, these fatty acids do not let ions out. So we have this accumulation of a proton gradient. So you have these protons getting pumped into the thylakoid space, as well as the protons from splitting of water. So you get a bunch of protons building up in this thylakoid space, establishing a concentration gradient. You have more protons, a higher concentration of protons within the thylakoid space compared to the stroma, which is outside. Now, it's due to this concentration gradient that we get chemiosmosis, the flow of ions through ATP synthase. So as those protons flow through ATP synthase by facilitated diffusion or through a protein channel, this ATP synthase turns and does what's called photophosphorylation. So the protons will flow through ATP synthase, turning, it kind of turns like a turnstile, and that will produce ATP from ADP and inorganic phosphate. So we actually will call this photophosphorylation. Now it's this ATP that will be used and invested in the anabolic pathway of the Calvin cycle. So the light reaction, in order to produce ATP, is dependent on this concentration gradient that was mentioned in Unit 2's standards. Okay, so now that energy captured, so it's also important to remember, hopefully you watched my playlist on Unit 3, but energy, when it's moved within a cell, it's moved in the form of electrons and electron carriers. So this NADPH is carrying energy over to the Calvin cycle. This ATP produced is also carrying energy over to the Calvin cycle. So the Calvin cycle is not mentioned very often in our standards, so you don't have to stress too much about all the intricate steps, but let's look at a general overview of how the Calvin cycle occurs. So in the first step, we start with a 5-carbon molecule called RuBP that we see in the GIF is right here. Okay, so when it starts, here we go, RuBP. And then there's an enzyme called Rubisco. So Rubisco is the name of the enzyme that will help do carbon fixation. So we see up here at this step, carbon dioxide from an air, like a gas of carbon dioxide molecule, is going to be attached to your Rubisco. So your 5-carbon Rubisco and your carbon dioxide will join together, forming a 6-carbon molecule that is very unstable. So right away, it kind of splits into 2 3-carbon molecules. So you see the split right here. Now, that first step is called carbon fixation, the actual taking of carbon dioxide from like the air and incorporating it into an actual molecule. Now comes the reduction phase of the Calvin cycle. So here we're going to invest that ATP that was produced during the light reaction, as well as take those electrons and hydrogens from that electron carrier, NADPH. So our NADPH is going to be oxidized, right, in this step. And our ATP is going to be invested as well. So the Calvin cycle is an anabolic pathway or an energy requiring pathway that builds. So one of the molecules that is produced, the main purpose of the Calvin cycle, is to produce this molecule called G3P. So G3P is what comes here. So this glyceraldehyde 3-phosphate. G3P is a very simple 3-carbon sugar, and 2 of those will form glucose. So other G3P molecules can follow other metabolic pathways and form lipids and amino acids, etc. So this right here, the Calvin cycle, is basically how you are taking air and then incorporating it to form organic molecules, which form the basis of life. So when we say life is built from carbon dioxide and water, the water component is this hydrogen and electrons from that electron carrier being invested. And that's the Calvin cycle. Ooh, so now we move into aerobic and anaerobic respiration. So when we look at cellular respiration, we're basically looking at 2 options of how to produce ATP. So just by looking at these 2 GIFs, you can see there is a difference between anaerobic and aerobic respiration. Aerobic respiration requires oxygen, and anaerobic doesn't. You also see they both start with glucose, which is the red G. So they both start with glucose. In the cytoplasm, the blue area, you can see how that glucose is split into 2 pyruvates in both of them. You can also see that in that cytoplasm, so we are looking at like this area here, in both of them, ATP is produced during glycolysis. However, in aerobic respiration, you'll notice that these pyruvates, look right here, are going to enter into the mitochondria to continue to be fully oxidized, to get any remaining potential energy from those bonds that it can. But in that mitochondria, it's going to require oxygen. So you can see the input of oxygen flowing into the mitochondria. And the waste product of aerobic respiration is going to be carbon dioxide and water. Okay, let's go ahead and study these. So in cellular respiration, in eukaryotes, we have a series of coordinated enzyme-controlled metabolic pathways to harvest our energy from our food. So basically, how the plants just invested that ATP and those electrons and hydrogens in the anabolic Calvin cycle, we are now taking that energy by breaking those bonds and removing those electrons and hydrogens, the opposite of photosynthesis. So our 3 pathways we're going to look at is first glycolysis, followed by the Krebs cycle and the electron transport chain. Now, the Krebs cycle and electron transport chain are only happening in aerobic respiration, so we'll also discuss what happens out here in the cytoplasm following glycolysis if it's anaerobic conditions. So first, glycolysis is a series of 10 steps, all using enzymes that basically are going to start with our 6-carbon glucose. And as you can see, the glucose has a lot of bonds that can be broken. And as we break down glucose and we take those electrons, that is called oxidizing our food, or oxidizing our electrons, our glucose. We are hoping to completely oxidize our food, harvesting all the potential energy from it. So in the process of glycolysis, we end up with 2 pyruvates that are 3 carbons each. We get 2 ATP and 2 reduced electron carriers. Now, if this were fermentation and anaerobic, that's all the ATP we would get. However, if it is aerobic respiration, there is still potential energy left in these pyruvates. So we're going to need to harvest that energy. So our next step is called the oxidation of pyruvate, where we're going to start in the cytosol. The pyruvate that was made in glycolysis occurs in the cytoplasm. This will travel into the mitochondria. So what we're seeing here is, let's see if I can play this again. So we have our pyruvate. It'll be oxidized, forming a carbon dioxide and a reduced electron carrier. I'm going to go again. So here we're going to have our bonds will be broken. A carbon dioxide will be released. That is 1/3 of the carbon dioxide coming out of your mouth comes from this step. And then we will have our 2-carbon acetyl, which are these 2 gray right here, will attach to coenzyme A. Coenzyme A will help it attach to a 4-carbon molecule called oxaloacetate. So this 4-carbon oxaloacetate will join with acetyl-CoA, forming citric acid. Now we can begin the citric acid cycle. So in order to actually begin the Krebs cycle or citric acid cycle, you need the oxidation of pyruvate to happen first, and that occurs within the mitochondria. You get 1 reduced electron carrier as well as a molecule of carbon dioxide. This is going to happen 2 times because there were 2 pyruvates made at the end of glycolysis. So now you have your acetyl-CoA is going to join the oxaloacetate, and as we go through the Krebs cycle, you can see that it's a series of, in the beginning here, we're going to oxidize, which means we are going to break another bond and reduce an electron carrier. So you have your 6-carbon citric acid. You're going to oxidize or break a bond, and that comes off as carbon dioxide, which is this right here. And we get a reduced electron carrier. And that happens again. So then you're down to your 5 carbons. And then a carbon dioxide will be oxidized, broken off, and then a reduced electron carrier. Now, as we go through the Krebs cycle, we will produce, so here you have an electron, a reduced electron carrier, another reduced electron carrier, another reduced electron carrier, and a 4th reduced electron carrier. So in the Krebs cycle, or the citric acid cycle, the main purpose is to fully oxidize our food. By the end of the Krebs cycle, our entire 3-carbon pyruvate is now in the form of carbon dioxide. You had the 1 carbon dioxide from the oxidation of pyruvate, and then you had a carbon dioxide produced here and here as well. So now you have 3 carbon dioxides. So at that point, your pyruvate is completely exhaled. Now, in the process of the Krebs cycle, you do produce 1 GTP that will be used to make ATP by substrate level phosphorylation and 4 reduced electron carriers. So you can see as we go through glycolysis, the oxidation of pyruvate, and the citric acid cycle, we are reducing electron carriers along the way, trying to get as many as possible over to the electron transport chain so we can make a ton of ATP. In glycolysis, we had just 2 ATP made. The Krebs cycle, we had 2, 1 for each pyruvate. But it's really going to be the oxidative phosphorylation that produces the most of our ATP. So when we look at the electron transport chain, the electron transport chain takes advantage of the 2 membranes of the mitochondria. So the mitochondria has 2 membranes, thanks to the endosymbiotic theory. And what we see here is in between these 2 lipid bilayers, you get a proton gradient. All of these protons accumulate between these 2 lipid bilayers because they're positive ions. They cannot diffuse out. So we get this electrochemical gradient, or a difference in charge across the membrane. So how does that happen, right? This is a bit, let's go. I guess let me clear the ink and we can draw on this and make it more smooth for us to understand. So here you have these electron carriers that were produced or reduced during glycolysis and the Krebs cycle. When they arrive at the electron transport chain, which a lot of them are already produced in the matrix, the Krebs cycle happens in the matrix of the mitochondria, and that's kind of where this is already occurring. So we're going to oxidize the electron carriers. So you see here, this NAD+ can be reused in glycolysis, the oxidation of pyruvate, or the citric acid cycle. Now the hydrogen that was on this electron carrier is pumped into this intermembrane space, and the electron is passed down the electron transport chain, where oxygen, which is very electronegative, is pulling and attracting those electrons down the ETC. So when oxygen gains those electrons and attracts them, it will also pick up some protons, some hydrogens. Now, that is how water is formed during cellular or aerobic respiration. So, as the electrons are passed down the electron transport chain, with oxygen being their final electron acceptor, they pump protons into this inner membrane space, establishing an electrochemical gradient. Now, it is that high concentration of protons that we call the proton motive force, with all of these positive charges wanting to repel each other. They actually will flow through ATP synthase. It'll turn and phosphorylate ADP and inorganic phosphate, forming ATP. We call this process oxidative phosphorylation. So this is kind of a intense picture. But when we look at here, the electron transport chains can occur in chloroplasts and mitochondria, as well as the prokaryotic plasma membranes. So we saw the electron transport chain in the light reaction earlier, but it also is occurring in the mitochondria. So in cellular respiration, what we have is we can see that the electrons are delivered by NADH and FADH2, which were formed in the previous metabolic pathways leading up to the electron transport chain. In aerobic respiration, the final electron acceptor is oxygen, and this is why we breathe. In photosynthesis, it was NADP+, we saw at the end of the light reaction. And basically, the transfer of electrons helps form that. I'm just basically summarizing everything we talked about. Forms the proton gradient across that inner mitochondrial membrane, which separates and creates a high proton concentration compared to a low proton concentration. And that will allow for the diffusion of ions, or chemiosmosis, through ATP synthase, which will produce ATP. And so that flow of protons back through drives the formation of ATP from ADP and inorganic phosphate. So in the mitochondria, we call this process oxidative phosphorylation, and in chloroplasts, we call it photophosphorylation. I want to emphasize, though, in plants and autotrophs have both chloroplasts and mitochondria. So this is occurring in both mitochondria and chloroplasts within plants. So it is not true to say, oh, plants have chloroplasts and animals have mitochondria, but rather autotrophs have both, like plants will have both mitochondria and chloroplasts. So just because it's talking about a mitochondria and oxidative phosphorylation, do not automatically assume it is only occurring in an animal. Look for other context clues in the question or the problem. Now, we also will use this cellular respiration. When we do cellular respiration, these metabolic pathways actually produce quite a bit of heat. And that heat is what we use to help maintain our homeostasis of body temperature. And so endotherms, sometimes we don't necessarily need all of that ATP, but rather we just need to generate the heat. So what we can do is we can decouple, which basically means undo, like disconnect, oxidative phosphorylation from the electron transport chain. So what that means is you can create like a proton leak that allows the protons to just diffuse back through into the matrix without making ATP. So it's really just a way to allow for metabolism to occur to generate heat without the production of ATP. And endotherms will use this to help us maintain constant body temperatures. And so if you think about our metabolic pathways, we are burning through our food, our glucose, like our carbohydrates, our lipids, and our proteins. So therefore, endotherms will require more calories than an ectotherm or a cold-blooded organism. Cold-blooded organisms do not use metabolism to generate heat for body temperature, and therefore they will require less calories in life for survival. So in periods of like food scarcity, an ectotherm would be able to survive longer than an endotherm of the same size. Okay. Oh, now we're almost done, guys. So fermentation. In fermentation, what we have is we have following glycolysis. Now, if there is not oxygen, right? Well, first off, let's look what we've learned with aerobic. So here you have glucose. And that glucose went through glycolysis to produce pyruvate. And we took the NADH, NAD plus that was oxidized, was reduced here into NADH, and that was carried over to the electron transport chain to make a lot of ATP. However, in the electron transport chain, that NADH will be oxidized back into NAD+. So therefore, we can have that that NAD+ can be recycled and used again in glycolysis or in the Krebs cycle, etc. However, when oxygen is not, when oxygen is not present, and we are in anaerobic conditions, we need to make sure that the cell does not run out of NAD+. So the purpose of fermentation is basically to oxidize this electron carrier and recycle the NAD+, so that at least glycolysis can continue. Because in anaerobic respiration, it's these two ATP. Those are the only ATP produced, and that cell is struggling to make ATP, and therefore glycolysis cannot stop. Otherwise, the cell will die. There'll be no more ATP. So therefore, we have two options of fermentation. In this example, this is lactic acid fermentation. You can see here in this GIF that basically in anaerobic respiration, we are converting the NADH back into NAD+ to continue glycolysis. So it is a cycle that will produce lactic acid as its product. So here you start out with your glucose, and then you have your NADH was made during glycolysis. And at the end of glycolysis, those are your products. However, in order to continue glycolysis, we need to oxidize these two electron carriers back into NAD+ to continue continuing this step of glycolysis. Without this step, glycolysis would stop. So therefore, you have to make sure that you have NAD+ to continue. Now, the other type of fermentation, I don't have a picture of, is alcoholic fermentation. And in alcoholic fermentation, basically, instead of lactic acid, you would get carbon dioxide. Oh man. You would get carbon dioxide and ethanol. So, sometimes in fermentation, if the clue is that carbon dioxide is produced, then you know it is alcoholic fermentation. And if it's lactic acid, well, then it's lactic acid fermentation. Okay. And our last slide here is it says the conversion of ATP to ADP releases energy, which is used to power many metabolic processes. But when we say conversion of ATP to ADP, ATP is actually hydrolyzed. So it's through the process of hydrolysis. So right here in this step, when that third phosphate is broken off, it's by the addition of a water to break that bond and hydrolyze ATP, releasing that energy to power cellular function. All right, I hope this Unit 3 PowerPoint was helpful for you, and good luck on your AP test or on your unit test. Great job.